Hi, everyone. I'm Joshua Pickard. Thank you for the introduction. I'm a student in DCMB, like you were saying, in rajapakse's lab. Today, I'm going to talk about the Hypergraph Analysis Toolbox, which is a toolbox of a bunch of different methods we have been working on in the group, some predating me, some we're still building out. And the methods are all the theme is we can do a lot of different things with hypergraphs. So I guess today, I'm going to tell you about a bunch of different things we can do with them, not following any one specific data set or problem, but sort of showcasing on a few different examples what we do and what the general methods are. These are ideas I'm very excited about. I've sort of been stewing, learning about hypergraphs, thinking about these for like the past 3 years. So I'm really excited about them. I like it a lot. If you have any questions during, just call out, or after. I'm like really into this. So with that, I guess let's just get going. So to give like a little bit of an outline of what I'm going to talk about or how we're going to frame this, I'm just going to begin by giving some general background of what is a hypergraph, why am I so excited about them, why I think you guys should also be excited about them. And to frame sort of the story or what motivated our group beginning working on these, I'm going to talk a little bit about Horsey data, which is one type of chromosome conformation capture technique that allows you to capture multi-way interactions, or it gives one multi-way picture of how chromatin is folded within the nucleus. Then after I give a little bit of motivation for why hypergraphs are so exciting, I'm going to talk about a few different things we can do in the toolbox, of different ways we can represent the graph, how we can compute different distances and similarity measures, how we can do some basic tensor entropy and other tensor-based calculations. And then I'm going to talk a little bit about dynamical systems on hypergraphs, which is one of the areas I'm most excited about. And lastly, I'll just say like a little bit about some current projects we're doing to continue this work. So with that, let's get into it. So hypergraphs are a very simple idea. The idea of a hypergraph is hypergraphs are networks where edges contain more than 2 vertices. So if you've heard of graphs or network science or like graph-based neural networks, traditionally the idea is you have a set of vertices, and typically your vertices are going to be the variables you're interested in. So if you're looking at RNA-seq, your vertices might be the genes you're interested in, and then you're going to have edges, which are going to be pairwise relationships between the vertices. So and like a gene regulatory network, your vertices are going to still be your genes, and your edges are going to be which genes can regulate or co-regulate one another. And those edges can have different weights. They can be like activating, they can be repressing. You can have a bunch of different sort of graph setups. But many types of interactions you can't describe exclusively with pairwise interactions. That's where the hypergraph comes in. Instead of 2 genes regulating one another, you might have 3 or 4 or n many genes that are all regulating each other together. And because of that, we have one hyperedge that's going to describe how they all interact with one another. So hypergraphs are this idea that was originally comes from math, but they're being used in a lot of communities in bioinformatics, in ecology and supply chain. Recently, I was reading this paper about how Amazon is trying to use hypergraphs to improve their search algorithms. So it's one thing that we're going to talk about Horsey data, but it's a general idea that we can fit sort of many different types of data or many different problems to. So I think there are 3 values of hypergraphs, and in short, that we can have more exact representation. The dynamics on hypergraphs very naturally fit like nonlinear systems, and that they're very efficient to work with from a compute standpoint. So to talk a little bit about why they're helpful for representation, let's just think about, suppose we have 5 people. We have Amy, Bob, Carl, Dan, and Ed, and they're going to email one another. So let's say Amy emails Bob, Carl emails Dan and Ed, and then Amy emails Carl, Dan, and Ed. So in a graph setting, our vertices are going to be people, and then our edges are going to be who emails who. So if we go to the graph case, we're going to say, oh, Amy and Bob were on one email here, so we can draw Amy and Bob together, one edge between them. And then we can say, oh, we know Carl, Dan, and Ed all email each other, so we have edges between Carl and Dan, Dan and Ed, Ed and Carl. And we can do the same thing for the 4-way email. And if we look at just this picture of the graph representation, what we're going to be able to see is we're going to know who was definitely emailed at the same time as one another. We're going to miss all sorts of information about what the community structure is. If we preserve the multi-way interaction, where here we have the same vertices, we have the same 5 people, but now we're gonna capture each relationship with sort of these colorful hyper edges, which are shown to like contain multiple people in the same email at a time. We're able to see, or we're able to preserve the structure of Carl, Ed, and Dan were all emailed together. And by keeping it in this structure, we actually have more information than if we go to the graph structure. So from like its most simple reason why hypergraphs matter, well, if we go to the just regular graph case, which I think is a little bit more common, you lose all sorts of information. So the other advantages of nonlinear dynamics, I guess I'll talk a little bit about more in a bit, and then time and memory efficient. The reason they can be very efficient to work with these graphs is because storing the sparse information or storing the hypergraph structure where you have the multi-way edges as opposed to the pairwise edges, you actually have to store fewer hyper edges total. Here we have to store like 3 edges. Here we have to store more than 3 edges. Oh, so we're going to save a little bit of space in our computer, which can make it faster to compute some stuff. So to talk about or motivate like how our group got into working with these, I want to talk a bit about chromosome confirmation capture. And the idea of this is if you have your chromatin unfolded in the interphase nucleus and you want to understand how the chromatin is relates to one another and what regions are near one another, you can do this class of techniques. And what you get is you'll get some type of picture of which loci were near which other loci. And the idea began, I think, in 2002 or 2003, where you could capture pairwise interaction. You could say, well, this one loci was near this one loci. And in 2009, Hi-C came around, and you could do the whole genome-wide pairwise interactions. Where here in this contact matrix, I'm going to use a pointer, I guess, maybe, or I'll just point. Okay, in this contact matrix, the vertices, we can think about it as a network. The vertices are going to be the different regions of chromatin. And then the weight inside of the matrix is going to be how often those regions of chromatin were observed as being in contact with one another. And we can think about it as some type of network where our vertices are loci and our edges are going to be interaction frequencies. And it captures like a very nice pairwise picture. The multi-way or one multi-way analog of Hi-C is Por-C. And there are other multi-way analogs, but I guess I'll talk about Por-C because that's the data we have in our group. And Por-C follows a similar process to Hi-C, where we begin with the interphase nucleus and we have our chromatin sort of floating around to some free strands. And the idea is we're going to take these regions, we're going to freeze the chromatin, we're going to fixate it, we're going to digest it, so we're going to cut off or sort of snip out these chunks here. And then we're going to connect all of these chunks together so that when we sequence it, these concatenamers are going to show what are the regions that were initially connected to one another in the nucleus. And by doing this, sort of we're going to see, oh, these 2 regions, we're going to have one edge between this vertex and this vertex. Here we're going to have a 3-way edge. These 3 are all near each other at the same instant. And there we have a 4-way edge. And similar to the email case, we could go ahead and say, oh, well, since we have this 3-way edge, 3-way hyperedge, we really just have 3 pairwise hyperedges. And if we stored that information, we could store it in a matrix just like this, which people refer to as like virtual pairwise, where really you extract a multi-way contact and then you store it as pairwise. But when we do that, we're gonna have the same issue of the email case of were these 3 all together in the same instant in time, or were they observed separately as being pairwise related, but there's no multi-way interaction. So Por-C allowed us to extract the multi-way interaction or the multi-way contact at the same time. And because of that, if we want to keep that information, we have to represent it as a hypergraph. So here we have sort of the same figure on the left describing this process of, oh, we have our chromatin and we extract these 4-way contacts. Now let's talk a little bit about, okay, what does a hypergraph structure look like? So here we have a bunch of vertices, which are going to be different chromatin loci. And we have a bunch of multi-way contacts extracted from Por-C data. This is what's called sort of the incidence matrix representation, where the rows are going to be your vertices or your chromatin loci, and your columns are going to be your multi-way contacts. And there's going to be an entry in like the I row in the J column if the I loci is observed in the J chromatin contact. And this is sort of the most sparse storage of your incidence matrix of your hypergraph. Here's sort of the same set of contacts. We do this virtual pairwise expansion. we're now, rather than storing multi-way interactions, we store each pairwise interaction. And when we do that, we have many more edges rather than hyper edges. And now we can store it similarly in this incidence matrix, or we could do like a high C adjacency matrix type thing. But either way, however you store it, it's going to take more space to store in your computer, and it's going to be less precise because you're not going to be able to uniquely say which regions were together in the same instant as opposed to which ones were just being observed in year one of it. So the work we did with this, or one of the projects we did with this data, was we took porosity data, we constructed a set of hypergraphs from the data, and then we wanted to see, okay, when we extract these multi-way contacts, can we actually learn anything interesting about it? Is there actually some regulatory structure going on that we can observe uniquely with porosity that we can't observe with high C? So what we did was we took our porosity data and then we filtered it down using a bunch of ATAC-seq and RNA-seq, and we looked for transcription genes that are co-regulated by the same transcription factors. We found a bunch of clusters of genes where we know each of these genes, or the genes in these groups shown here, all co-localize in the nucleus, meaning they were observed in the same porosity concatemer, and they have a similar regulatory mechanism as one another. And we were able to find this because we preserved the higher order contacts. But if we remove the higher order contacts, we went just to pairwise, then we would be able to say, oh, we know it's possible that these genes are near each other at the same instant in time. And we know it's possible that they have the same regulatory mechanism, but we couldn't say definitively that regulatory mechanism was acting on them simultaneously. So this is like one of the projects we did with the porosity data. Something coming soon is we're in the group we're working on doing single cell porosity, where we basically do the same technique on single cells. This is a project that one of my lab mates, Cooper Stansberry, is very involved in and very excited about, and shared the slide with me so I could tell you all about it so everyone can get excited about it. But I don't have too much there yet, but we're gonna have a lot of hypergraphs once we finish with this data. Currently, we're struggling, or not struggling, but changing some ways we align it to see if we can just get any more information out of it. Okay, so that's a little bit about what hypergraphs are, why I think they're important, because again, they're precise and they're efficient to work with, and a few instances of what we can do with porosity with a hypergraph representation. But porosity is by no means the only area we can apply hypergraphs. Kan Chen is one of the students that used to be in our group and is now at UNC, and he's been doing a lot of work on metabolomic networks with hypergraphs. This is from one of his papers, I forgot the citation. People have been looking at hypergraphs to understand the structure of the brain and how neurons are interacting with one another by analyzing MRI data. And also people have applied them to ecological networks to understand how different species regulate one another. So these are just a few instances of other areas we could bring this idea. Okay, so that's, I guess, a little bit of an overview why I'm excited about hypergraphs and what they are. And now I'm going to talk a little bit about what we can do with this toolbox. But again, if you have any questions, or it's not clear what a hypergraph is or why you should be excited, please call out with a question. Yeah?
So I know a lot of the graphs you've shown in previous slides are like directed graphs.
Mm-hmm.
Is there like a directed version of a hypergraph?
So that's a great point. So here we sort of have arrows denoting how these edges are going to flow. We can see from A and B, we flow to C, D, and E. There's some notion of directionality. In the porosity data, we're extracting that all of these chromosome loci are near each other at the same instant in time. There isn't so much directionality associated with it. We could look at, and we've thought about looking at when we read the concatemer, first we read one loci and then another, and trying to use that to associate like some direction with it. But we haven't been able to do that so much with like when we were trying to identify the transcriptional hubs. There we can say a little bit that there's a hyper edge or a directed hyper edge because we know there's particular transcription factors that are co-regulating all of the same genes. So maybe we could say the hyper edge is the genes that are seen together as well as the transcription factors, and those are being directed at. But like with most porosity data, before we do that, we're saying it's undirected. But for like some of the dynamics, definitely people define the directed version of this. Yeah.
Yeah, so no directionality, then I don't know if there is such thing, but like can you add like weights to the hyper edges?
Sure. So directionality and weighting are two things we're glossing over. You can add both directionality and weighting to them. In like high C, the weighting is clearly just the number of times that contact is observed. So when we look at this as like an adjacency matrix, we see there's like the intrachromosomal contacts are like more brightly colored red because we know there are going to be more contacts there. And we think of that as being like the weighting in our network, where if there are more contacts of high C, we know that those loci are closer to one another, and we just call that the weight. We could do a similar thing for porosity, where we say, oh, if we observe one concatenator many times, we could assign it a weight similarly to this. One issue or what way that can be a little bit more challenging is with high C, we're only looking at pairwise interactions, and the set of pairwise interactions is much smaller than the set of multi-way interactions. So each concatenator is more unique. So it's tougher to assign a weighting to it, but we could do that as well. Or for example, with like a network, a reaction type network, or with the reaction network, you could also assign weighting as different rate constants for how fast or slowly your reactions will progress. And lastly, also about the directionality of these networks. I think the first example is this email network of like Amy and Bob and Ed. There would be very natural to say, oh, we have a directed hypergraph where one person is the sender, everyone else will be the receiver. So you have some notion of who sent it, where it starts, where it goes to. So yes, we have directed and weighted versions of these. Another thing that I didn't mention so much, but is also important, is what's called a uniform hypergraph. So in a graph, all your edges are going to be pairwise. Every edge will have exactly two vertices. In a hypergraph, you can have a hypergraph where every hyperedge contains a variable number of vertices. Like if we're looking at 4C data, okay, each concatenator is going to contain between 2 or like 8, maybe on the larger end, chromatin loci that are observed at the same time, and that's what we call non-uniform. But if we have a graph or a hypergraph where all hyper edges contain 3-way interactions or 4-way interactions, we call that uniform. And that becomes a little bit important for how we represent these as tensors. So I guess that's actually a good segue into like, oh, how do we actually represent these hypergraphs? So this is a figure that we made sort of outlining all the different things we can do with this toolbox. And now I'll go through and talk about them each a bit. So here's is, I believe, the instance of the same hypergraph I had in the cartoon model of generating 4C data. And we have sort of 5 vertices, and we can pretend there are chromatin loci or whatever other variable of interest. And we have 3 hyperedges, and we can say those are our 4C concatenators or whatever other type of relationship we want to say we have among our data. And we draw it as a hypergraph where the edges are going to denote where the colors are going to denote the different hyperedges. And this is the instance matrix, which I think we talked about a little bit before, where the vertices are going to be your variables or your nodes, or the rows are your nodes, and the columns are going to be your hyperedges. And for uniform hypergraphs, we like to think about how to represent these as tensors. And in the matrix case, or the graph case, where it is really like a 2 uniform hypergraph, every edge in a graph is exactly 2 vertices. Here we have a bunch of different graphs. We think about the adjacency matrix as being one of the main ways we represent it. We represent numerically the graph structure. And what goes on in the adjacency matrix is here we have vertices 1 through 4, and there's going to be a 1 in the ij entry of your adjacency matrix if vertex I and vertex j have edge or are interacting with one another. So here 1, 2, and 3 all interact with vertex 4. So basically the column in row 4 are dense with 1s. In the hypergraph case, or the uniform hypergraph case, we're now, instead of having pairwise interactions, we can have 3-way interactions like here. We're gonna have an adjacency tensor, which is just the multi-way analog of the adjacency matrix. So here we have a 3 uniform hypergraph. Every hyperedge has 3 vertices. So now our adjacency matrix becomes an adjacency tensor. where we have rows, columns, and then maybe slices going forward and back, and each one is similarly indexed with vertices. And for instance, this blue square here is going to be indexed with V4, V2, and then, oh, it's in the front slice, V1. All right, so blue is V4, V2, and V1, and the hyper edge here is going to be, the blue hyper edge above is going to be on vertices V1, V2, and V4. So that's sort of how we get these like tensor representation of the hypergraph. Another thing we can do if we have the hypergraph is we can still represent it as graphs, like we talked about before, where we could say each multi-way interaction becomes a set of different pairwise interactions. So here above, we have another hypergraph on 5 vertices. And what happens here is we can do what's called the click expansion. And in the click expansion, you have your same vertex set, and each of the 3-way interactions just becomes 3 pairwise interactions, like we see, or like what we talk about with doing like virtual pairwise from Porcy. If you take your Porcy and you decompose it into all the pairwise contacts, you get like it almost looks like high C data. And it's nice because we have the same vertex set in this click expansion as we do with the initial hypergraph, but it's problematic, like we talked about, because you're actually going to lose some information about your system. Another type of way we can represent hypergraphs as graphs is we can do what's called a star expansion. And what goes on the right side here is we make a graph representation of the hypergraph where we actually increase the number of vertices. So now we had 5 vertices and 3 hyperedges in our hypergraph. And in the right side, we're gonna have 8 vertices, 5 representing hypergraph vertices and 3 representing hypergraph hyperedges, which are gonna be the ones on the left. And we have sort of an edge in our graph between the vertex and a hyperedge representing vertex whenever that vertex is contained in the hyperedge. And what's nice about this is from the star expansion, you can uniquely recover your hypergraph, which is very nice. You never want to lose any information in how you're storing your data, but it's problematic because now our vertex set is different. So if we go to actually draw this as, or if we were to like draw the adjacency matrix of what this looks like, it would look nothing like the virtual high C or the adjacency matrix we get with this structure. So from the hypergraph, the takeaway from the slide is we can represent it as a few different graphs. We can represent it as the tensor. The tensor is very important. So we're going to compute all sorts of different higher order SVDs and other tensor properties from it. So one thing we can do with our toolbox is we can compute different hypergraph similarity measures. So if you have your hypergraph, the most natural question whenever you have your data is, oh, what is the distance between my data points? If we have Porcy from 2 different cell types, we might want to compare or ask ourselves, how is the structure of the chromatin in these cell types different from one another? And we can do that sort of a few different ways. One way we can do it is sort of the simplest is we can take our Porcy data, we can do this virtual pairwise expansion, we can make it into like a regular graph, and then we can look at different distance metrics on the adjacency matrix of the graph or on just different graph measures. Another thing we could do is we can make it into the star graph, which is even better because now it's going to preserve all of our structure, but it's going to have this awkward issue of now we have a new vertex set. And then the third type of measure we've been working on is we could take the tensor representation of our data and compute different tensor distance metrics. The tensor one is nice because it preserves in the best form the multi-way structure, but it's problematic because if you have non-uniform data, if you have Porcy data, and you want to do some tensor-based distance metric, well, you have to find some way to make your non-uniform data fit into your tensor. So sort of what's shown on this slide is we generated a bunch of synthetic hypergraphs using like standard random hypergraph methods. And then we have each of these different distance measures where we compute it on the click graph, on the star graph, and directly on the tensor. And we have Hamming and spectral and centrality based distances. And what is shown in each of these plots is each color is going to correspond to a different type of random hypergraph we made. So we made some that are totally random, some that have scale-free properties, some that are small world. We make a bunch of different hypergraphs and we try to compute the distances between each of them, and then we project it with T-SNE so you can see what's going on. And we see with some measures like, if we do the star expansion and then we look at Hamming distance on it, there's very nice separability between all of these graphs. With other measures, if we do tensor-based Hamming distance, this would not be good. We wouldn't be able to see how the data is different from one another. So, sort of a part of this project and all the things we compute is just propose or look at different ways we can compute these measures. And even though in this case, a star Hamming is probably the best one up there, you have to choose the distance that'll be appropriate for your data. So on these random networks, that one works very well, but on different types of networks, you want to use different distances to discriminate.
So can I ask you a question on this?
Oh, yes, please.
It looks like in this case, the click is not much worse or even better than the star. Is that right?
Oh, yeah, click Hamming is good. Thank you for pointing that out.
If that's not a hypergraph approach because it's making all the edges, do you have a synthetic case where using the hypergraphs is actually beneficial?
So that's a great point. In this case, the star expansion, the click expansion, you can both discriminate the data very nicely. And then the synthetic data shown here, certainly not. Certainly, to do a click or star would give you the best discrimination. One thing I would say, well, like one caveat where you're saying is, the graph-based approach works just as well as the hypergraph-based approach. It's true because of the click. That it works well in the click graph, but it's not true because of the star graph, because the star graph is still preserving the hypergraph structure. So, like talking about the click graph as a graph, both of these are graph objects, but the star graph object is still representing the hypergraph structure. But in this case, I think like, that they're definitely like work as well as one another. I don't know, does that make sense?
Yeah, I mean, I guess if you just go to like the normal graph rather than the hypergraph, in this case.
Yeah, in
you
this
don't
case,
need
you'll do
hypergraph.
just as well.
So I'm curious if you had a synthetic example where the hypergraph actually outperformed not using a hypergraph.
We don't have a synthetic example for it. We have, like, I guess when we've looked at a lot of this 4C data, we've found that, the hypergraph case works better, or the star expansion case is the best. The reason I say the star expansion case is the best is because in the synthetic example, the click does very well. In the not synthetic, in some of the real data, we've seen the click has not done as well. The star expansion, in my mind, is preferable for the tensor representation because even though we can store these tensors very sparsely, computing like spectral measures on these tensors with a large number of vertices or high order is a little bit expensive. Yeah.
We can talk about it.
Okay, yeah.
There's a question online from Gil, and Gil, I think I've given you the ability to unmute yourself. Do you want to ask your question?
Yeah, I think, can you hear me?
Yes.
Yep.
Great. So this is a very striking slide, Josh. I didn't quite understand your explanation. Normally, with Tisney, we want to see as separated foci as possible. But I think you said that the star method with overlapping of 2 of the 3 subgroups is somehow more informative than the click method. Would you try again to explain the star versus click?
Sure. So I'll use a pointer maybe so you can see it on Zoom, if I can get this to work. It's the bottom button. Oh, top.
Yeah,
button.
I see it. Yeah, I see
Okay,
it.
so I'd meant to say the star Hamming distance, the one on the left over here, is as
Yes.
good as the click Hamming distance, the one above. The star, well, were you referring to the star spectral?
Yes.
Yeah, so the star Hamming and the click Hamming distances perform about as well as one another, because like you said, we're able to see a separation between the clusters.
Right.
Um,
And
the
then why do that? What is the spectral? Is that a different experiment altogether?
So the spectral distance is a different type of graph distance that we can compute on the star graph or on the click graph. So it's gonna look at what are the eigenvalues of the click graph versus the star graph. or in the tensor case, the tensor eigenvalues. And the reason we show all 9 plots here is because with this synthetic data, the star Hamming distance and the click Hamming distances do the best. Those are the distances where we see maximal separation between the synthetic data. The reason we investigated these other or the remaining 7 distances is because with other data sets we found that some of the tensor distances perform better than the star or click one, and you have to choose sort of an appropriate distance for your data.
Is it possible that some of these methods are showing separation that's not real? Not biological?
In this case, it's all synthetic data.
Yeah.
So to explain maybe the experiment more clearly, we generated hypergraphs according to 3 different models. We used an Erdos-Renée model where you randomly, on a fixed set of vertices, you randomly choose hyperedges. We generated them according to the Watts-Strogatz model, which is a variety of a small world network, where you begin by sort of making a lattice on your hypergraph, and then you randomly permute which hyperedges are attached. So you're going to have some attachments that are going to go across the lattice. And then the third model is going to be a scale-free hypergraph, where what we expect to see here is a degree distribution that will follow some type of power law. And then on these hypergraphs, we wanted to discriminate and see if we were able to find distances that discriminate them well. And the star Hamming distance and the click Hamming distance perform best in this case on these synthetic data. I don't know if that answers your question.
Yeah, I'm just trying to understand what the physical meaning is of these clusters. And maybe that's not what's being shown here.
Well, what's being shown here is if given the hypergraph, we're able to predict or separate out which model was used to generate it. For instance, and the problem we would want to do if we were looking at data in this experiment rather than these synthetic examples, say, given a set of hypergraphs or given pore C from different cell types, can we discriminate only from the pore C data which type of cell or what was the process that contributed to that hypergraph we observed? In this case, I think it's tough to say what if there's like a physical meaning behind the distances. We're just trying to discern which model of graph it is.
Okay, thanks. Thank you for the effort. I appreciate it.
Maybe we can talk more about it after, yeah, but thank you.
Sure.
Okay, another thing besides distance we're interested in is to compute entropy of different hypergraphs. And entropy is important. We've looked at it. We've looked at hypergraph entropy of the organization of this chromatin conformation capture data from fibroblast cells throughout both the cell cycle and cell reprogramming. So the reason the tensor representation is important is because when we compute tensor-based entropy of these hypergraphs. And the way tensor entropy calculations work is we're given a tensor up on the left T, and we compute some type of higher order singular value decomposition. And from the singular values, we then compute like standard entropy with Shannon entropy. And what's shown in the left is on the top, if we begin with and 7 vertices on the top and 7 on the bottom, and we want to add hyperedges in a way that is going to maximize the entropy of the graph, we can follow the procedure on the top. And in the bottom, if we want to add hyperedges that are going to minimize the entropy of the graph, we can follow the procedure on the bottom. And one thing we did with our data was we took hypergraph representation of the chromatin structure of fibroblasts throughout the cell cycle and normal proliferation. And what we tried to do was compute the entropy at different time points. So we had 8 different time points. And on the left figure, we show the tensor-based entropy, where the blue line is going to be the entropy of the reprogramming fibroblast, and the orange line is going to be the entropy of the proliferating or the fibroblasts just going through the cell cycle. And here we, on the right-hand side, we compute the same thing, we compute graph entropy. So we take the hypergraph representation, we compute entropy on the click expansion of the network. And one argument we tried to make, or we make that was made in this paper, is that when we look at the tensor entropy, we're able to sort of distinguish these two processes slightly better. Yeah, that's the idea of, I guess, tensor entropy. But we've, I guess, tried it on a few other data sets as well.
Sorry, can you explain how you can see that that's better discriminated on the left than the right?
That's a great question. I think it's sort of a qualitative argument. It's not, I don't think it's a good argument that it's better discriminated. One thing we could say is we look at the scale of the change. From here, it's about a shift of 0.4 in entropy. Here, the largest shift is maybe about 0.02. I guess it's a larger change.
Is there any replication here? I don't see any errors on the data.
We only had one replicate of this data, so there's no error bars, and it's probably not the most compelling. I will give you, it's not the most compelling instance of this.
Okay.
Yeah. Okay. One thing I've been working on lately is thinking about like dynamical systems that occur on your hypergraph. So if we have a hypergraph where we have these vertices, which are going to be our state variables, our variables we measure, and we have hyper edges, which are going to be the interactions in our network. We can take our adjacency tensor and sort of write down the homogeneous polynomial of the adjacency tensor, and we can look at what are the controllability or observability properties of that system. So, and sort of maybe the best example of this is down here. I have a graph on 3 vertices and I have a hypergraph on 3 vertices. And this graph is going to have some adjacency tensor A, the hypergraph, or well, this will be an adjacency matrix. This will have an adjacency tensor. And if we write down, I guess, sort of the classic like linear dynamics on a network, we're going to get x dot equals ax on the graph. And in this case, we're going to have some linear differential equations where the derivative of x1 is going to be x2 plus x3. In the hypergraph case, rather than having linear equations, we're going to have nonlinear terms, and those nonlinear terms are generated by the homogeneous polynomial of the tensor. And what we really mean by that, or one way we can think about that, is we replace every addition with multiplication. So this term, x2 plus x3 will become x2 times x3. And we're going to have nonlinear dynamics. And from these dynamics, we can think about how can we control the system, how can we observe or make some type of sensor measurement from the system. So this is a project I did not do at all, but talking to a system is controllable if it can be driven from any initial state to any target state. And in the graph case, if we have these linear dynamics, determining if a system can be controlled from a set of input nodes is a classic problem where we form what's called a controllability matrix. And the idea of that matrix is we can give it a very simple test where we just look at the matrix rank, how many columns are independent of one another. And when the matrix is full rank, we're able to characterize if we're able to control the system from the set of driver nodes that we have access to. This is an important problem because if we have some type of system and we want to say what are the minimal inputs we need in order to control the system, we want to have some procedure for identifying those driver nodes or those variables we have to modify. Rather than having the linear case, we have these nonlinear dynamics of the hypergraph, and now we want to be able to ask the same question: how do we identify the minimal set of driver nodes that are going to allow the dynamics to be controllable? So we have sort of an algorithm for doing that based off computing like repeated SVDs of the adjacency tensor of this nonlinear controllability matrix. We're able to evaluate sort of how different input signals are going to propagate over the network, and then say if this nonlinear controllability matrix is full rank, similarly we're going to be able to control the system. Here we have this isn't a great data set, but I have a few different graphs that are shown up here. And this is not a good figure. This is not a good figure. But what's going on in this experiment was we had a bunch of time series data, and we fit polynomial models to this data. And from those models, we said, oh, we have this hypergraph structure, and the hypergraph structure was formed according to the polynomial. We found the tensor whose homogeneous polynomial was a polynomial we fit to the data. Then, given these different hypergraphs, what we want to do is we want to say, if we look at the hypergraph representation versus the graph representation, how does the number of driver nodes that are required to control the system change? And what we saw is for the connected component, always the number of driver nodes required is going to be smaller than the number of driver nodes when you have a linear model to your data. This is flawed in part because the graph isn't always connected, because we were trying to enforce a sparse constraint on our model, or on the model we fit to the data. Another reason it's flawed, or it could be flawed, to say that we need fewer nodes to control the hypergraph system is because, oh, well, if we fit a linear model, we fit a nonlinear model to the data, still we're only fitting a model. So the ability to control the system is going to be predicated on our assumption that our model is actually a good representation of the system. But it was interesting to see that these dynamics are easier to control in the nonlinear case.
Do you have an example of where you would use these types of models?
For the observability case, I think it makes a bit more sense to say, yes, I do.
Would you mind sharing it?
Yeah, let's do it. All right, so, okay, here's where we're gonna use this model. Suppose we have these same nonlinear dynamics, we have these polynomial on the adjacency tensor. Now, instead of controlling it, because this is my project, the control is done before me, but the observability one, I guess, is what I'm working on now. So, and we say similarly, the system is observable if the states of all nodes in the hypergraph can be uniquely determined from the sensor nodes we have. So if we have n variables, we're only able to measure m of them over time. Or what we want to be able to do is to say to pick the best m variables that are going to allow us to estimate the state of all other n variables. And we think this is important maybe for biomarker detection, where we want to find the minimal set of sensors that are going to allow us to estimate or observe the rest of the system. So maybe from talking about the polynomial model, I don't know if that'll be more convincing or to talk about the data, but in these polynomial models, we have a bunch of graphs in this figure. And what we did was in these graphs, we're gonna have linear dynamics. And we use this observability matrix, which is similar to the controllability matrix, to say, what are the minimal set of vertices that we would have to monitor over time in order to make the system observable? And we drew sort of one arrow towards each of these vertices. Then what we did was we added a single hyperedge to each of these graphs to say, now we have a mixture of three-way hyperedges and pairwise vertices. And we did the same procedure again to say, what are the minimal vertices that are going to be required in order for us to observe or make some type of estimate on the system? And then we added in a bunch more hyperedges again. And again, we asked, what are the minimal set of vertices that are required to make some type of estimate on the system? And as we added in more hyperedges or more higher order interactions, the number of vertices required to make the system observable, and the dynamics are changing from linear to a little nonlinear to very nonlinear. The number of nodes required to observe the system decreases. And what we've been looking at is the data we've been looking at for this is sort of time series RNA-seq collected during just normal fibroblast proliferation, as well as time series RNA-seq collected during fibroblast reprogramming to myogenic lineages. And we're trying to think about different ways we can use this sensor selection approach to find what are the most important genes that we have to be measuring during these time series experiments. Because if we can reduce the set of all genes we measure to a very small number, we would be able to capture that at a higher frequency or temporal resolution, which might be helpful for making some type of estimate about the system. Still, maybe it's not that compelling. You're gonna say, oh, it's only a model, but we think if we can fit these nonlinear models to it, and we might be able to find smaller sets of sensor genes that are important to observe. Yeah.
Would it be possible that in any given model, all these relationships actually exist? For example, you may have linear relationship past non-linear relationship across different platforms.
So there are two things there, to repeat the question. So the question is it possible that all of these different types of relationships can exist across different time points? We have linear relationships, we can have nonlinear relationships, and these can change over time. Definitely it's possible, not possible, probable. Like certainly, the relationship is gonna change over time. We've been working on how we can do linear and nonlinear relationships together in the same instance to select observable nodes. And we've also been working on temporal linear relationships. So if you have linear relationships that are changing over time, and now the next thing like I want to do is to think about, oh, if you have these nonlinear relationships that are also temporal, how we can model that. And the thing that's shown here is, I guess, if we select different sensor genes, how well we can estimate other cell cycle genes. And this is, the idea is like, can we actually apply this? Can we find some subset of genes that are going to be informative for estimating the other gene? And in this case, the different colored lines are going to correspond to the number of genes that we're measuring. I think ranging from like 1 to 50. And the black dot, and again, these aren't beautiful, plots are the real data. And what we want to see, or we want to see over time, is the estimate converging to what the true measured value is from based off only estimating the sensors or only looking at the value of the sensor genes. And then I think my last slide is talking about how well we can actually scale these models. And one issue among many with these tensor-based models is the scalability of tensor calculations. If we want to look at the controllability of the dynamics, we have to compute higher order. We have to compute a variety of a higher order SPD. If we want to look at different distance measures or entropy measures between hypergraphs, we have to compute, or one way we can do is by computing tensor eigenvalues or tensor decompositions, which can be very expensive if we have like a high dimensional data or many different high order interactions. So I think the last thing is I've been looking at different ways we can factor the tensor using some type of Kronecker factorization and then improve the calculation or the time it takes to calculate different tensor eigenvalues and tensor decompositions. I think this is my last slide of just the people who are involved in these projects. Cooper helped me a lot with building the tool that does all these calculations we have. He helped me a lot writing the code. Ken used to be a student in my group as well, in Indika's group, and now recently just started at UNC. And then Professor Block, Amit Saran, and Indika were involved in helping make all these methods. Yeah, that's what I got.
Questions?
Yeah.
You said at the beginning that there was some computational efficiency with the tensor representation, like less storage. Did you see that in any of your practical applications that was a beneficial effect of the method?
So in terms of like the efficiency in terms of tensor calculations or tensor storage?
I mean, I guess you said that one of the reasons why you might want the hypergraph representation is that it's more compact.
Yeah,
And that might lead to computational efficiency.
sure.
Did you see that was an important factor in any of your applications?
In almost all of our applications, the speed at which we can these calculations take a long time. And it was always an important factor to have both the most sparse storage as well as the most sparse representation of using like sparse tensor formats. So yes.
Vaguely, but no
Yes.
quantitative assessment.
Yes, we didn't really do a quantitative assessment to see how much slower it would have been, because we sort of tried a few things and we're realizing it's slower. Like we saw it, so we saw there was an advantage to doing it, and we never benchmarked how much faster it was. Okay. All right, yeah.
In your example with emails, you don't have any false positives, and in certain types of data, you won't have as many false positives. But in C, due to chemistry, it can happen that sometimes you observe certain regions being close, even though they're not actually close biologically. So the frequency of like how often we can find certain lesions together is important to know as a threshold after which we can say, okay, this is actually biological interaction, not just artifact. Do you have any false positives filters or some other reading that you do in Toolbox or not?
We don't have any false positive filters. One of the plots, or one of the things we thought about with the entropy based calculation, say, oh, as we add more hyper edges, how is that going to change the entropy of the system to see if one hyper edge is going to cause like some very unique change and all of a sudden it's going to be very different, or whether that won't be the case. We do have some code for saying, given this set of hyper edges, how can we predict new hyper edges? Which is going to be the opposite problem you're talking about. How do we say, given this, how do we find false positives? We don't have any methods for that.
So for your modeling, would you be able to also calculate some regular properties that the regular graph network would usually come up with, that regular neighbors, for example, like a centrality or small workers?
Centrality or small worldness or clustering coefficient or diameter. Yeah, I guess maybe I should have said that we can calculate we have code to calculate all of those as well. And one other thing about the tool, I guess I should just tell you to plug the tool, is we wrote it in both MATLAB and Python because we wanted to just really make sure we were getting the same answers between both. So you can use it in either language. Yeah, it's easy to download, so everyone should check it out.
You should attach the link.
Oh, I should link on the side. Okay. Yeah. All right. Thank you all for coming.